I would just like to introduce you shortly, and then I let you talk about your research interests. So we have Dr. Don Cleveland. He is chair and distinguished professor at the Department of Cellular Molecular Medicine. He is working at the University of California, San Diego. And he won many, many scientific awards, among others, the Breakthrough Prize in Life Sciences, which he won in 2018. And Dr. Cleveland has made seminal discoveries in how cells divide, and also what drives the development of neurodegenerative diseases, and actually also how to cure them. So we will hear a little bit about that. We also have Dr. Michael Sheetz as a guest. He is the director of the Nano Medicine program at the University of Texas Medical Branch. He also won many, many awards, and he has actually a tremendously productive career so far. And as I understand, this is now a next phase, his new position. So we're also going to hear about his research a bit, and he has pioneered a field that actually didn't exist until he arrived at the scene, and that is mechanobiology, and he will certainly talk about this. And he won the Lasker Award for this research that he did in collaboration, and he and his colleagues were honored for some of the work, not on biomechanics actually, that was a later achievement. So, but anyways, and last but not least, we have Dr. Michael Elowitz, professor of biology, bioengineering, and applied physics, so multidisciplinary position at the California Institute of Technology, and he's also an investigator of the Hughes Medical Institute. And Dr. Michael Elowitz really probably represents a field of the future that is probably just at the cusp of a breakthrough also into medicine. And in 2007, he was the winner of the so-called Genius Award. And he won this award for his design of the first, or one of the first synthetic gene control network. So he called it the repressilator, if I am correct. And what this did, it helped initiate yet another field, and that is synthetic biology. So I think we have a very interesting program. So a very warm welcome to you, and thank you very much for participating in this event. And so I just want to start maybe again with Dr. Don Cleveland and ask him to give a little bit overview of where he came from and what his research interests are.
So I started my scientific career. I always knew I was going to be a physicist, and I indeed went to graduate school in physics. But then I became interested in biology, and I switched out of that, and I never looked back. That was one of the best decisions I ever made, and I ended up purifying a protein which became a famous protein in neurodegenerative disease, tau, which accumulates inside Alzheimer's disease. And then with the discovery of the genetic forms of major neurodegenerative diseases, we just started testing. We didn't know much about them, but we just started testing models for how they worked. We showed that the causative genes caused one or more toxicities. And then we thought, well, if that's true, why don't we just turn off the bad gene? And then we set out to try to develop a strategy to be able to selectively change expression of individual genes within the nervous system. And we succeeded in that. We now have one successful approved therapy in a childhood disease. We have 7 ongoing trials in the major diseases, 3 in ALS, 1 in Huntington's, 1 in Alzheimer's, to suppress tau, the protein I found a lot of years ago, and 2 that started this year in Parkinson's disease.
So, and what does tau do as a protein?
Oh, that unhappily, that is still not an answered question. So we thought, I think our community thought that it induced the assembly of tubulin into microtubules. But when you delete it from a mouse, the microtubules are still there. With tau-free mice are amazingly normal. They're not completely normal, but they're amazingly normal. So the idea that it was stabilizing these neuronal microtubules might do that, but it's not essential for that. So what the normal function is, I think, actually has not been resolved.
So you will have the drug before you know what it's doing.
So some say, actually, some of my colleagues have said that that I've given up on science because I say, but I know in the examples where mutations are causative disease, we know the mutant protein does one or more bad things, and Huntington's is an example where the same mutant gene causes every case of Huntington's, and yet there's our community, the research community, there are 15 different models for what mutant Huntington does bad. And I'm thinking, my guess is that in reality, probably everybody's right, that they have a piece of the toxicity which is seen in multiple contexts. But you know what? If you could just turn off the bad gene, you'd hit them all. So if you could do that, so I thought, well, let's just do that.
So that is actually a surprisingly common theme in gene therapy, that targeting complex diseases that one doesn't really understand how they arise in many cases is actually very successful at the moment. We also think about cancer, immunotherapy, and other therapies. So that is very interesting. So one doesn't really need to know before one can actually do something about it.
Yeah, but although I'll let you should move on to the others. But I say, but we do know it is, and in examples of what we think of as non-genetic examples of neurodegenerative disease, like the most instances of Alzheimer's, most instances of Parkinson, we still know that proteins accumulate, normal proteins accumulate, and that if individuals express those proteins at higher levels, they will get early onset disease. So too much of a good thing can be bad. So I would argue there that while we don't know the details downstream of that, we do know mechanistically that too much synthesis of a normal product in many cases, and certainly of the mutant products in other cases, that's directly the founding event. So we're going directly at the disease mechanism.
Great, wonderful. So Dr. Sheetz, I would like to ask you the same question, maybe introduce your research focus a little bit.
I mean, I started out as a mathematician and chemist, and then was interested in medicine, and so moved into biology. And basically followed a number of problems. And we were initially interested in the mechanics of membranes, and then got into the motor proteins. And that was very productive and a lot of fun. Then with the laser tweezers, we moved into membranes, again with forces. And that led us to really the first understanding that mechanics really make a huge difference. And most recently, we've been able to define the way in which cells sense their environment, or at least one way they sense their environment. And that way, which is a rigidity sensor, and like my pinching here, pinches the matrix and thereby measures the rigidity of the matrix. And it's missing in all cancer cells that we've been able to define. And surprisingly, as a result of that quote unquote transformed state, the cells become mechanosensitive. And so we formed a company to mechanically address or treat cancer using ultrasound, and that's what we're really about. One of the major things we're about now. in Galveston, and the preliminary results on that are very promising. So it is indeed just, I think, the tip of the iceberg in terms of how we can use mechanical manipulations to affect aspects of the human body and the human condition and improve, in this case, cancer. We have preliminary evidence on aging as well, where mechanical aspects seem to be very important.
And this is also a process that is critical for stem cell differentiation, no? So
as well, right. Basically, we don't, as Don was saying earlier, our understanding of a lot of these processes lags behind our ability to manipulate them.
Yeah.
So, and I think that going ahead, and it's certainly a major area for the future in terms of the students, to just understand how the organism is formed during development, because it's a very mechanical process, and it's essentially deterministic.
And you really only know that this process exists because you somehow managed to visualize many of the processes that, because there's very localized activation of signaling and also dynamic signaling. So you actually had to develop many of the tools. So I guess your background may have helped.
Nanotechnology, it's not our nanotechnology, but nanotechnology of our engineering colleagues has been extremely helpful in all of this.
Yeah, but Michael, wouldn't you say that that's actually, that's the way science really works. We're always borrowing from someone else, and that the great discoveries are made by someone borrowing from here, borrowing from there, and then seeing how they can put all of that together. So.
And that is actually a theme in all of your work, basically, because you really collaborated also with people of different backgrounds, and neither of you, and also our third speaker, has really a biology background. So maybe that is actually helpful, because you're not bogged down in details. You can ask the simple questions that many times people in the field do not ask.
Sure. New perspectives are very important.
Yeah.
Yeah, they are the ideas. So when we first moved into neurodegenerative disease, it was after the first gene was discovered that was causative of ALS. It's a gene whose product works in oxidative chemistry, and the entire medical research community said, "Hey, the mutation." They're going to lower the function, and it's a loss of function mutation, and that you got dominant loss of function. And I looked at those mutations, and my student and postdoc looked at those mutations. My big team, there were 3 of us, and we thought those were point mutations. They could all be lowering the activity, but it's not. But actually, there were no null mutations that would really lower the activity from that gene, and it thought, they should be there. And so then we tested, and we made 10 or 11 of them. We showed that they all had a range of activities from 100% retention of normal activity to 0 activity. So well, that doesn't make any sense. And so again, to your students, that's just it. So the experts knew the answer, but the answer was not right.
That's actually where you tell your students that they should ask the dumb questions.
Yes, they should. Absolutely. Always ask the dumb question.
So coming back to Dr. Sheetz, you're now in Texas. So you had quite a long career in Singapore. So you've actually seen many different environments. So what is your, nano medicine, that is interesting. What are your plans in Texas?
Well, as I was saying, the use of ultrasound, which acts at the cellular, subcellular level, to really cause the death of all cancer cells that we've been able to test so far, opens up new ways of approaching that. that really don't, I mean, the standard approaches look at the issue of how do you make a tumor and what can you do to inhibit tumor growth. Our approach is to say, what is the sensitivity of these transformed cells, and how can we kill them directly? And surprisingly, they seem to be mechanosensitive. And that approach, under conditions that don't hurt normal cells, and in fact regenerate senescent cells, will cause the death of the cancer cells.
But also tissue stiffness is often a earlier stage of metastases, so it basically is pretty much late in tumor development, but before metastases. So if you could visualize stiffness and differences in stiffness, wouldn't you have also a relatively easy, non-invasive diagnostic tool to look at cancer?
Right. I mean, for us, the idea here is that the ultrasound is not damaging normal tissues. So we are building a quote unquote spa in which you can sit with the ultrasound treatment, and you should be able to kill all metastasized tumor cells. So, you know, this is not proven, and indeed, we have a company, blah, 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 all the caveats should be added here. Yeah. Everything at the basic level supports that idea.
But wonderful, you just started your position, so you have hopefully many years to go and develop new tools and therapies. So wonderful. So I want to move to our third speaker. So Dr. Elowitz, I think when I looked at your research, I realized that the theme of this session, the universe inside the cell, is actually a bit too narrow, because all of you actually look at a more complex system and cell interactions and cellular behaviors in populations. So at least you do now, using relatively simple model organisms, though I believe you also wrote that you want to go into the brain. So maybe you can tell us a little bit what's on your mind.
Sure. Yeah. I mean, I would say the universe inside the cell, I think, is not too narrow in a way, because the universe outside the cell is created by cells and through the circuits that they have, circuits of interacting molecules that are operating inside of the cell. So I think I'm okay with the universe inside the cell. I think that's. And
you're not a biologist either, right? Your background is outside biology as well.
Oh yeah, that's right. So maybe I'll say like. I got my undergraduate degree in physics, and then I went to Princeton, also in physics. But with the idea that I would do cosmology, which I was really excited about. Yes? Sorry,
no, I was going to say, did you finish in physics? Because I went in physics, but I didn't finish. That was it. Yeah.
You're right. I think maybe like you, like my Jedi physics training was incomplete in some fundamental way, because what happened to me was I just saw at Princeton that there was amazing work going on in the physics department, but a lot of it was based on building really complex detectors, high energy for high energy physics and things like that. It was really giant projects with huge teams, big engineering challenges. And then one day I walked across the street and I was talking to Stanislaus Leibler, who was a theoretical physicist, and he had just decided to try to set up a little lab in the corner of the biology building. And going back to the discussion of asking different questions, I think, Stan is just kind of really embodies that. So he came in and was trying to ask very different questions than the biologists were asking.
Well, they didn't put you into the cellar, so.
Actually, it was a nice lab, actually, third floor. And actually, my very first project in the lab was working with kinesin, the protein that...
But you destroyed it, no? Your colleagues here started studying these proteins, and you tried to develop methods to destroy it.
That's
No?
true. That was like
So of course, destruction is also Dr. Cleveland's goal. Not that protein, but
not that protein. Yeah, not those proteins. Those are
So can you tell us a little bit about what you did to modulate kinesin, Dr. Elowitz?
Sure. I mean, maybe I could just, I think that, for me, I think the main thing that I've been really interested in, starting with kinesin, but now kind of going through bacteria and then into mammalian cells, the main through line for me has always been, I guess, just this amazing idea that if you look at the cell, it's like an incredible little device that can do things that no other device that we know can do. So they can copy themselves, they can replicate, they can communicate with each other, differentiate, to go and develop into a multicellular organism, and then even create an immune system in that organism that can control it and eliminate disease. It's just, there's nothing like it. And I think what the question really is, well, where do those amazing capabilities come from? And for me, the most interesting level, and everybody has a different level, so it's just personal, is the level of the circuit, the level of multiple genes and proteins interacting with each other to generate these behaviors. And, I think for me, I just got fascinated by the idea that, if we could understand those circuits, they're kind of a little bit above the level of pure chemistry, but they're not quite at the level of like, what is life really altogether? They're sort of in between. And if we can understand those circuits, then we could understand all these behaviors. We could control cells better. We could program cells to do new things. And that has been kind of the thing that I'm chasing, I guess, all these years. And for me, the first step actually came in grad school in Stan Leibler's lab, when, I had been reading a lot of biology papers, and at the end of every paper, there was a model of some system with a bunch of arrows on it. And I just wondered whether, instead of trying to infer those arrows from experiments in which you delete a gene or add a gene or whatever, whether we could actually build a circuit from scratch. And that led me to try to think what would be the most, basic, important, fundamental circuit I could think of that I could actually build. And that was the repressilator that you mentioned in the introduction, which was a simple oscillator that was designed to make cells blink on and off. Just 3 repressor genes hooked up in a negative feedback loop, kind of like a game of rock, scissors, paper. And I think so. So that, I think, being able to do that, I think for me just was like, oh, look, we can actually build a circuit in a cell and program that interesting behavior. And that led to a whole series of other things, trying to think about where does the variability in that circuit come from. That led us to questions about noise and stochasticity. Yeah. And then more recently, now what I'm really into is we've kind of shifted the lab into mammalian cells. And we started getting interested in the potential to engineer therapeutic circuits. So these are still circuits, but they're circuits that can do something that no individual gene or protein could do. And also try to, at the same time, on just the fundamental side, ask, can we build circuits that replicate some of the more fundamental, fascinating behaviors that we associate with development, the ability to maintain different stable states and switch between states and communicate between cells and things like that. And so my lab, what we try to do is kind of have a balance between programming circuits based on ideas of how we think they should work, and on the other side, trying to learn new ideas about circuit design by looking at natural circuits. So we'll also try to analyze communication pathways, epigenetic regulation, and things like that.
But it's still interesting because you study randomness and noise and how at some level this becomes non-random, deterministic, right? So it's actually very interesting and sometimes troublesome if you think about your brain, about what's going on and how you may make decisions and form thoughts and memories. So I really wonder whether maybe you can also start with my next question. And where do you see the capacity of this research going, let's say, not maybe in 5 to 10 years, because you talked about this, you're going to do that work in part yourself, but maybe beyond that, longer term. What do you see? You talked about circuits, therapeutic circuits. So what do you think is going to happen?
In the coming decades, you mean on the circuit side of the therapeutic circuit? Well, I guess the way I think about it, like, I think in medicine, we've been dominated for a long time by the paradigm of a drug. A drug is a molecule, and the molecule can be very specific for another molecule, and drugs are great. But if you think about a lot of the really exciting things going on, and I would include Don's amazing work on in gene therapy, also regenerative medicine, also cell therapies like CAR T therapies, these are all paradigms that are based on taking advantage of the function of the cell, programming the cell to do something, something new. And I think to me, what is exciting is the idea that the cell can think, or the circuit in the cell can make decisions. It can assess the situation, coordinate with behaviors with other cells. It can just do things that no individual drug could really do. And so when I think like kind of what's out there in a decade or 2 decades, it's like, well, can we ask, what would it take to actually be able to design a therapeutic circuit that allowed a cell to go in and, essentially, let's say, recognize a tumor or discriminate a tumor cell from non-tumor cell, or other kinds of disease states, and actually kind of process that information and do something about it in a program, but not just like maybe it'll work, try to snap it together and hope it'll work, but in a more rational and predictive way, where we can program these circuits and we really know they're going to work the way we think they are. So I think turning biology into this kind of forward design science, and that's happening. I mean, there's a huge explosion of synthetic biology right now. It's just taking off, especially on the mammalian side. And so I don't think that's a unique vision, but I think I think it's exciting. And I think it's also something where, as different people start to solve different parts of the challenges, whether it's on the engineering side of like being able to, engineer circuits faster and, assemble DNA and deliver it and everything, or on the kind of fundamental understanding side, identifying the designs for these circuits that are actually going to function effectively in the cell. And maybe just to finish, that's where noise comes in, because one of the reasons noise is so interesting is that in an electronic circuit, noise has been designed out of the system and you just don't have to worry about it. But in biology, noise is really part of the system and the cell is using it. And so it's interesting to ask, how do we design our circuits in a way that is natural to the biological context, and how can we learn from the way natural circuits work when we do this?
Yeah, because cells do not receive normally just one signal from their environment. They receive many. So noise actually makes sense, because otherwise you would receive one signal and you couldn't really react to other signals. But if there is a level of noise, it depends a lot on how long you get a certain signal and how strong the signal is. Is it very localized, or is it over a certain region of the cell? So that makes a lot of sense. And if you think about hormone action, so circuits, I think in diabetes would probably play a huge role for patients, because then you would have maybe more personalized therapies rather than just injections and hope you get the eating wrong and nothing happens.
Well, Michael, the important thing that I think most people in designing those circuits have not taken into account is the mechanical aspect. And indeed, this rigidity sensor that I talked about earlier is in many ways the sort of device you're talking about. And indeed, it has inputs, outputs, and all those things that you would like. And it has been very difficult for many of the mechanical aspects. I mean, diabetes, for example, can be cured by exercise and diet. And, so is it better to use that approach or to try to engineer the biochemistry? I think going forward, we'll have a lot of interplay there.
But
right. Sorry, go ahead.
No, go ahead.
No, I was going to say, I just was going to agree with Mike that, yeah, sort of integrating mechanosensation into these circuits is really going to be critical. And so, you know, I think that's where it's, I think there's sort of two aspects of it that I think are interesting. We try to do both. One is sort of the core computational componentry that you need to be able to just process information in a generic way. And the other part is the interfaces that you have to have with the natural cellular systems, because your synthetic circuit is never going to act, or at least not in my lifetime, ever going to act alone. It's always going to be needed to read out what's the signaling environment out there and so forth. And it's also going to be able to need to actuate responses in the cell. So it really needs to work with the pathways that are there. And that's what's, I think, so fascinating to me, is that by trying to do something synthetically, very artificially, it forces us to ask questions about the natural pathways so that we can use them. We have to ask kind of operational questions like, how do we read out BMP signaling? Or how do we, control a cell death pathway in a particular way or something like that? And so it turns the natural systems, it just opens up kind of a different kind of question about them, right? Like, how do I use them? Not just how do they work?
But also like psychiatric disorders, depression, there the treatment is also give a relatively high steady level of medication, whereas the fluctuations in principle are way more important. So with circuits, maybe one can also address such diseases or disorders and help patients. So you can pulse the dosage instead of having a steady dose delivery.
Yeah, absolutely. I think the dynamics are really critical in these circuits and underexplored, probably mainly because they're harder to do. It's harder to control the dynamics, harder to read out the dynamics. Even amazing techniques like single-cell RNA-seq are usually still snapshots. Yeah, that is something we spent a lot of time on, is really looking dynamically at what these different pathways do and how do they encode information in dynamics. And that seems to be I think it's something that's very prevalent in biology, that the encoding is in time, not just in concentration. And if we only could look at temporal dynamics of everything at the cell at the same time, we would see that. But of course, it's still much harder to do that than to take a little snapshot. But I think
assays are hard. The assays are very hard. I mean, Don knows the dynamics of his favorite proteins are very rapid within cells. And we still don't understand why they are so rapid and how, nevertheless, you get a standard or quote unquote normal behavior.
So, but in general, what comes out is kind of a lot that is still not known, and also certain playfulness and curiosity that you all have or still have after long careers or mid-term careers. And I can really sense that. And I think that is also something that I wondered about. I mean, obviously, you are very gifted individuals, but my question that I also kind of sent you is, obviously you went to universities probably that kind of promoted such thinking. But if we now look here in our developing country, can actually curiosity and vision and critical thinking and creativity, can that be learned to some degree? Can we teach it? And if yes, what would be the best approaches? Because nowadays we still kind of separate the fields, and it seems to me you all came from different directions, and you had questions in biology that others may not have recognized. And sometimes we just drown students in facts and ask them to learn the facts. So this is the kind of old way of teaching and learning. But nowadays we have this explosion on data. And there is no way students can kind of keep up with the new knowledge. So looking back at your experiences, was there something special Was there a way of learning, or teachers or mentors that inspired you?
You've got three people who have very physical backgrounds here.
Yeah.
And I think the fundamentals of physics and chemistry are always going to be operative and are always relevant in all these systems. So training there is indeed needed. Training on the latest method to understand the stock market variations and the statistics thereof, et cetera, may make you money, but really doesn't understand the biology.
Well, these two fields are also what biology students often struggle. So they try to get rid of these topics as in the beginning of their undergrad studies, and if possible, don't go back. So that's actually wrong. So physics and chemistry should probably be more integrated throughout the biology, at least, curriculum. So
I think physics in particular, I'm very biased.
I think we all agree with our bias.
But I do feel that it leads you to at least want or expect or demand a deeper understanding of things, or a certain kind of understanding, and feel dissatisfied with superficial understanding. And so it doesn't mean that we always achieve the deep understanding, but I think it does give you a certain taste or something. And I think, to me, even now, when I have students or postdocs coming from physics backgrounds, it's like we're always a little more in sync on what's interesting. Not that they're better, worse, smarter, not smarter, but just that a little more, there's some similarity of taste, I think, that is I really value a lot.
So you can think in mechanics and relativity theory, and you kind of can bring both of these directions in.
Yeah, I think what I found disappointing is that a lot of the engineers who study mechanics in biological systems are satisfied with a descriptive answer rather than a mechanistic answer. You know, if you do this, then you get that. It doesn't tell you how it works. It doesn't tell you the underlying mechanisms and so forth. And I think perhaps the physics and chemistry interests embodied here are, you want more than that pure description.
Yeah, and I think also all three of you at some point have used microscopy. And I think their physics background also helps you quite a lot. And also in developing tools of how to modulate the protein function and activity and visualize it, I think. So yeah, you're right. So that's what students should really be integrating more, or we should integrate more into their curricula.
Well, I think for me, I think the students, they need, it's all the best is for the students actually to be interested, and so and the backgrounds that we all three of us had. I don't use my computational background very much anymore, but if I had that background today and we're joining biology, those tools are even more valuable today than they were before, and you'll have a series of experts, sort of experts, who can handle these large data sets, but actually all of us could learn that. And the students who actually have command of that ability to do that, they'll just be more empowered to then test the biological question correctly. And I do think some of those representations from some of the p-values that are published in the big journals every week, I think they just don't look like they've been calculated correctly. I think the p, instead of being for significance, it's really for preposterous. And but it just enables you. And you, again, you asked the question, Jeannette, initially was like, so I don't. I've known Mike Sheetz for many years, but actually I don't know where he was an undergraduate. And Dr. Elowitz, we just now, I think, met for the first time. But me, I came from New Mexico State University, not one of the elite universities of the U.S. And I think the preparation from a university preparation all over the world can generate students who have who are asking questions, and if you do have a tool set that includes not just the facts of so-called facts of biology, but who have tools, these computational tools that you can really now exploit these the kinds of sequencing approaches that have now been done, the kinds of modeling approaches that Dr. Elowitz was discussing, or the mechanics that Dr. Sheetz was discussing. You'll be really well powered, and many students who have more traditional, more pure biology backgrounds, they'll never be able to do that. They'll just be intimidated by, oh, I don't know how to do that, those large data sets. I don't know how to query that. And the answer is, of course you do. You just have to learn, and you just have to ask. And learning to ask. It's a lesson I learned from my... Very important, yeah, it's one of the lessons I try to teach students, and I learned it. I watched somebody, and when I was this beginning graduate student. I had left physics mid-year to join this biochemistry graduate program, having never taken a biology course. And I felt that I thought I was behind. I was just way behind. I was behind everything. And I took some undergraduate courses, and the undergraduates seemed to know everything. And I thought I knew nothing. And then I would go to the weekly seminar, and it was an era where everybody did go to the physical seminar. And there was this one guy, he was said to be the best and the brightest of the young faculty. And he sat in the front row, and every seminar he asked a question. And the students in the back, we were thinking, the question's just so simple. And so we couldn't figure it out. His questions were always so simple. And we couldn't figure out how he could be the best and the brightest, because he didn't seem to know anything. And that individual was Bruce Alberts, who was going to write one of the most world famous textbooks, and who became the president of the National Academy of Sciences in the United States. And Bruce taught me, and I had the great fortune to be adjacent to Bruce when I was a graduate student and also when I was a postdoc. And it was about halfway through postdocing when I realized the reason Bruce asked those simple questions was because if you had been able to do that, you would have really done something. Yeah. So it doesn't have to be fancy and big words, very long sentences. No, just can you really do something? And if you can describe it in simple terms, that's all the more powerful.
Yeah, and also knowing whom to ask for advice.
But don't be intimidated. Ask lots of folks. And I saw that. I've seen that multiple times where actually the men are typically more crippled because they just can't ask. And the women are able to ask. And I've seen some truly the world leaders who just were never afraid to look like they didn't know. And they would just ask. And there was one example where an individual asked me a whole bunch of questions about neuroscience, and I knew she didn't know anything about it. And she asked me really simple questions. I probably spent 2 hours on the phone with her, and I then recommended that she talk to 2 or 3 other people. And if you talked to her the day before, because she had built a mouse that really told us a lot about sensory neurons, even though she didn't even know where the sensory neurons were. And but at the end of the day, she'd spent the day talking to people, and all 4 or 5 of us knew that she really didn't know much. But if anybody else talked to her the next day, they're going to think, how does she know that? So it was, it took me a long time. I knew that was the right thing to do, but it took me a long time to implement it, to be able to say, no, I don't know about that. Why don't you, can I ask you about that? So where are the sensory neurons? And so, yeah.
So I think you would love our students here. So,
well, if they're asking questions, they're doing the right thing. They are absolutely doing the right thing.
Motivation is a much better sign for success than pure intelligence.
Yes. Yeah, I was going to say, I've always counseled that, a pure intelligence, or if you're really in mainstream biology, great experimentalist. Oh yeah, pure intelligence, that's good. Great experimentalist, that's good. Neither are necessary nor sufficient. For me, the only thing that's really, really necessary is perseverance.
Yeah.
You just have to keep at it. There are so many bumps in the research world. So perseverance is, in my view, is essential. It's not sufficient, but it's essential. And I think, the inquisitiveness that Dr. Sheetz just mentioned, I think that's that too. If you're interested, you don't have to be the most allegedly most gifted intellectually. You don't have to know the most about it. In fact, sometimes not knowing is actually the key, because I probably indicated one of those earlier today, where the field thinks they know something that's been shown for years, and yet it wasn't really true what they thought was true. And the new folks, they don't know why the field thought the wrong thing, because they just thought, well, how would you know that? Why do you think that? And when you start thinking that way, you're probably right.
I mean, the other thing maybe to throw in there is that one of the great things about biology, especially now, is it's so huge and open. Yeah. And like, you could take any system, and I think this is maybe a lesson, sometimes the lesson that we can draw from something like CRISPR, which has been so revolutionary. I think the lesson we should draw is that fundamental research on just curiosity-driven, on interesting systems can lead to amazing, catalytic discoveries. Sometimes I think people draw a different lesson, which is we should just do more CRISPR. And that's the whole lesson.
Yeah.
but anyway, but you can, no, I'm just going to say, I think the point is, you can, I'm not sure how important the system is. Every biology is so rich, so diverse, that you can probably find something really interesting and important in almost any corner you look, I think, possibly. And so I think it's really, and it's a setting where, maybe the first one or two questions you ask, people kind of already know, in a particular system. But by the time you get to the third or fourth question, you'll probably hit the edge of what people really know pretty quickly. So I think it's a field that is very, or should be very welcoming and very open to students everywhere, because there should be a place for everyone to find their own scientific identity, the kinds of questions that they like, the kinds of systems that they like to work with, or kinds of problems they're interested in. It should be very open in that way.
I mean, one trap that it's important to avoid, when I was a grad student at Caltech, a lot of the brightest people in the lab would design an experiment and then figure out why it wasn't perfect. And then they would not do the experiment because it wasn't perfect. They didn't have all the controls, blah, blah, blah. And they couldn't necessarily interpret the outcome. But from my own experience, those unknown results that really are important.
Mike's completely correct about that. There are those, certainly I bet all three of us have seen this, where we have individuals who really seem super talented and they can really think through all this, and they actually have such a broad command of literature, and yet they're stuck with, they don't make the great discoveries because it's not really that perfect. It won't really do that. And I'm thinking, don't let the perfect be the enemy of the good. And because most of the things that were even the limitations of what would be perfect were based upon our current understanding of really how things are. And that understanding is not as perfect as we want it to be. So even when you say, I can tell you that that's not going to be a perfect experiment. Actually, you're not actually so sure of that either. And an incomplete experiment that gives you a great new direction or potential direction. I mean, nothing wrong with that. So yeah, don't let the perfect be the enemy of the good.
I mean, it needed some, also mentioning CRISPR, there is this long story of labs who studied in a field that was god-awful to many mainstream scientists, so boring, there is no money in it. And yet they worked and they kind of came up with interesting ideas. And then it took some people from the outside who realized also the potential for their work. And then you have this explosion in knowledge, and suddenly everybody wants to be in this field. So I think for students nowadays, it's sometimes actually agonizing to choose what they should go into, because they think they don't know what the unknown is, so what are new areas, and what would you recommend them? Because thinking back when I had to make the decision, it was between botany and molecular biology, and I hated botany, so I went into molecular biology. But nowadays there is all these cool topics and really wonderful.
I guess I would say, speaking as a very indecisive person, I suffer from this a lot, and I can certainly understand, the problem. At the same time, when I actually look at people in my own lab over the years, and I just look at trajectories of people I know, it just seems to me less important than you might think it is. I mean, it's important what you choose, but it's not the only thing, because if you start two people at the same project, they will drive that project into totally different directions. Yep. This is what I find. And so it's not one choice at the beginning, it's a series of choices over months or years that takes you to where you're going to go. That's one thing. And the second thing is that biology is so much more interconnected now. I think maybe if many years ago, you might go to just a fly developmental genetics meeting and it would just be with other fly developmental geneticists, let's say. But now those people are really in the same meetings with everybody else. And I think that's a good thing. And it means that the tools you're developing, the discoveries you make are likely to be broadly interesting. And so there's many ways in which you can shoot from one field to another. Personally, I think I wouldn't stress about it too much. I would worry more about, is this a fun lab? Do I have a good relationship with people? Am I on the same wavelength with the advisor? Is it, am I enjoying it? It's the process, I think, that really matters.
Right. And then I actually completely agree with Michael. But one other thing is, if you're looking at labs that you would like to be able to join, also if they're senior people like some of us on this in this session, then you might want to look at what have happened to the folks who've worked with them before. So how enabling have they been for the careers of the people who worked with them? And because that's what you're hoping for, that graduate school or postdocing, those are the steps that you need to get to your final job. And some people have been amazingly successful in having a long series of successful trainees, and others not quite as successful. And so I would certainly look at that, ask the previous trainees what they thought about the lab, because you want to enjoy what you're doing, and you and then I don't think it really matters. I agree that you're going to help develop it into a new direction. You're going to take it directions. If you have computational skills, you're almost certainly going to use those. And especially if you're working with biologists who do not have those skills, you'll have things that they want, and you'll be able to take the problem in new directions. There will be synergism.
And have fun.
The other point, which perhaps we didn't emphasize enough, is that in graduate school, you two guys changed direction. I was in chemistry already, but spent a lot of my time in biology. And the reason I went to Caltech was there was no barrier between chemistry and biology. Almost a biology professor.
Yeah.
I think the programmatic aspect should encourage interdisciplinary activity.
Yeah, and collaborations and people who can give you new ideas and have a completely different thinking. Yeah. So I think we're at the end of the session. So thank you so much. It was really inspiring and interesting, and hope you visit Kazakhstan one day and look at the talent here. And certainly we are going to look at what's happening next week and also what's happening in your career. So thank you all very much, and I really, really enjoyed talking to you all.
Thank you very much. I enjoyed it a lot. So,
thank you.
Our pleasure. Thank you.
Bye-bye.
Bye.